Good morning, everyone. We are from Group 1 presenting on a mind map on the topic of cell senescence culturing methods. So first of all, let's get to know what is all about the in vitro signs of aging, or in another word, the cell senescence. So what is the morphological change that will happen during the cellular aging? So it will experience the increase of the cell size, nuclear size, number of multinucleated cells, number of cytoplasmic microfilaments, number of vacuoles in the endoplasmic reticulum ER and cytoplasm, and also the increase of sensitivity to the cell contact. We also experience the permanent DNA damage, inflammatory secretome, chromosomal instability, and there will be an expression change in terms of the loss of C4s in the senescent cell of week 138 cells, and continue with the EGF signaling, impair the downstream receptor binding, and continue or proceed with the reduction of IGF-1 mRNA production under the detectable level. So these are the morphological change during the cellular aging. But what are the biomarkers of aging so that we can know or determine the amount of senescence achieved in a culture due to the specific staining of the senescent cells? So first of all, we have the staining of Beta-Galactosidase, which we can specifically stain the senescent cells in situ. And moreover, we have the annexin A5, which can accumulate at the nuclear envelope during a replicative senescence, and the drug-induced cellular senescence in the primary human fibroblast.
Hello, my name is Ainur Hanani binti Rosmi, and my matrix number is 205428. Based on the mind maps here, I would like to present about the quantification of cellular aging and the concepts in culturing aging fibroblasts. But firstly, let's see how to quantify the cellular aging. The keyword here is the population doublings. But what does it mean by the population doublings? It actually can be defined as the numbers of times that the cell number is doubled. It is assessing the population doublings that the cells experience during the culture phase in order to measure the cellular aging. But in general, population doublings can be measured by plating a given number of cells and counting them after a specific amount of time, to be exact in a certain time. The age of the cells is increased by one population doubling. For instance, 1.0 times 10 to the power of 6 cells are plated and followed by a growth phase and a count of 2.0 times 10 to the power of 6 cells. But however, a more exact procedure is required for precision because even counts as I described above are rarely never reached. So the scientists are still in the identifying process in this situation. So the next one is the concepts in culturing aging fibroblasts in aging research. One of the terms that have been used is cumulative population doublings, which is CPD. So what is actually a CPD? It is actually a number of the times that the cell has been doubled. So it has been used to count all the cell divisions and is influenced by a number of biological factors. For example, the species maximum of the lifetime, the donor's age, the location of the biopsy, and the culture conditions. So, the senescence cultures are those that fail to reach confluence after a week. So, when cultures are unable to complete one population doubling over the course of 4-week period that includes the 3 consecutive weeks of refeeding with a new medium containing 10% of FPS, they are deemed to be senescent at the end of their replicative lifespan.
For signalization and subcultivation, subculturing should be performed upon 90% of confluency to prevent contact inhibition or transformations, and the cells are in the long phase of the growth. In synchronization, PBS is used to remove the residue of FBF that may inhibit the trypsins, and trypsin is used to digest extracellular message protein that are responsible for the attachment of the cell. And EDTA is added in trypsins to enhance the cleavage ability of trypsins to help weaken the cell adhesion in cell suspensions. And the medium should be renewed every 2 to 4 days because of the nutrients and space limitations, morphological changes, and accumulations of metabolic byproduct level. For cell counting is used to determine the cell viability, and triple blue exclusion is used as rapid test for gross damage, which is conveniently combined with determining of cell numbers. As the triple blue dye is permeable to non-wearable cell or deaf cell, whereas it is impermeable to this dye.
Okay, the other procedures for culturing aging fibroblasts, which are freezing, thawing, and also monitoring aging process. The third one, which is freezing, cells to be frozen should be in late log phase growth. Firstly, dissociate monolayers with trypsin, EDTA, and resuspend cells in complete medium. Next, count resuspend cells to determine viability and number. Next, gently pellet cells for 5 minutes. Remove and discard supernatant above the pellet. Resuspend cells in freezing medium and freeze immediately. And fifth, cells frozen at negative 80 degree Celsius for short-term storage and container filled with isopropanol to prevent the formation of damaging ice crystals. And lastly, transfer cells in cryovials to liquid nitrogen after 24 hours for long-term storage. And the fourth one, thawing, recovery of frozen cells. Firstly, remove cryovial from liquid nitrogen and place in a water bath. Agitate gently until thawed. Wipe vial with 70% ethanol before putting under hood and opening. Next, transfer cells to a sterile conical tube containing medium pre-warm to 37 degrees Celsius. Okay, fourth, centrifuge 5 to 10 minutes and discard supernatant containing residual DMSO. And fifth, resuspend pellet in complete medium and transfer to culture plate or flask containing the appropriate amount of medium. And lastly, place in incubator and check cells for adherence after 24 hours. And lastly, monitoring aging process through aging biomarkers Beta-Galactosidase staining. Alright, firstly, monolayers at low cell density are placed in PBS containing 3% formaldehyde for 5 minutes. Next, after 3 washes with PBS, stain cells overnight at 37 degrees Celsius in staining solution containing 5-bromo, 4-chloro, 3-indolyl beta-D-galactoside. And next, cells are then washed with PBS. And lastly, beta-galactosidase activity is monitored visually by scoring blue precipitate in the cytoplasm and photographed at 200x. Alright, 200 times.
So, aging cell culture, major issues and experimental design consideration based on in vitro cell culture model to study in vivo cell senescence. One of the effect of culture condition in the differences in terms of environment between in vitro and in vivo cell growth is the exposure to direct light. Fluorescent light can cause DNA double strand to break, and any photoactive components of the cell culture medium can be damaged when there is prolonged exposure to light. Other conditions that induces premature senescent phenotype are the use of hydrogen peroxide, third-butyl hydroperoxide, UV, and also gamma radiation. Aging culture studies to mimic cellular senescence are time-consuming, and it needs a lot population doubling to get critically attenuated telomere lengths to trigger the cell senescence. It is better to study one serially cultured sample of different ages rather than using different cultured samples and examining it at different time period. This enables the researchers to restart the study at a certain point if any problem arises in the culture, then re-examining the culture sample at different period, which can take a long time and waste of energy. Hence, it is important to freeze the cells in liquid nitrogen at various ages. So I think that is all from our group, from group 1. Thank you for watching.